All right. Well,
One
Mark.
second.
Oh, yeah. I can hear you if you can hear me.
Yeah, it's my,
Just a little pause here, everyone.
sorry, my microphone. I've got two different microphones and one of them doesn't work very well. Can you hear me okay now?
Oh, yes, yes. We can hear you just fine, Mark. So,
Okay.
if you can hear me, we're good to go.
All right, so where was I?
Well, we were talking
Uh,
about how something about the proteins that were
yeah, oh
overexpressed and
yes,
killed the
the
cell.
toxicity. So one of the problems was that mitochondrial proteins aren't ever supposed to be found in the cytoplasm normally.
Ah,
And so they can actually be toxic for a variety of different reasons. And you need to, you, the interesting thing is that the mitochondrial proteins that are normally encoded by the nucleus, their mRNAs are translocated to the surface of the mitochondria, and then those proteins are translated and imported into the mitochondria at the same time, which is called co-translational import. And so Marisol Coral-Dubrinsky is my supervisor
yeah.
now at the lab that I'm working in, and she pioneered the use of this, so attaching appropriate signals onto the mRNA such that co-localization or co-translational import would occur with these transgenes. And she found that that was the key thing that made this non-toxic and effective.
And would make allotopic expression possible then.
Yeah,
Is
so
it?
she was able to do it in fibroblasts. That was 2 years ago. She did a series of experiments in fibroblasts that gave very good results. She was able to rescue cells carrying two different types of mutations, one in ND4 and another in ATP6. And
Ah,
so from those results, it looked great. It looked like she had working allotopic expression that was efficient and well tolerated by the cells. But of course, given the history of allotopic expression, people outside of her lab had reason to be skeptical with the results, and so
sure.
we needed to reproduce those results. And so at the time, I was actually in Cambridge, England, in a different lab, and we talked to Marisol, and we had her send us samples of her cells and her vector, and we tried to reproduce the result in our lab there. And actually, it turned out that we were unable to, which kind of was sort of what normally happened in the field.
Yeah.
And so that was a little bit discouraging. But what we decided to do was there's a lot of differences from lab to lab, and it's hard to get the sometimes when you just read a paper or send emails, you don't catch every detail of the methods. And so we decided the best thing would be for me to actually go to her lab and try to reproduce her results in her lab. And
Okay.
so actually, that's what I've been
That's what
working
you.
on over the
Okay.
summer.
And how close are you to getting to reproducing those results, or have you had any success as of yet?
We've had mixed success. One of the
Ah,
difficulties is the original cells that she used, which we tried to do the experiments in again. They're quite a bit older now, and they're starting to senesce. They're not really behaving the
but
way that we would want them
they're
to.
aging. They're aging,
They are,
huh?
and so we've recently acquired some new mouse fibroblasts from Douglas Wallace's laboratory in the United States in California, and he has mouse fibroblasts that carry mitochondrial DNA mutations. And I have some cells growing right now that have been transfected with allotopically expressing vectors,
Okay.
and should be getting results in the next few weeks on those.
All right, I got a basic technical question here for you about getting genes delivered into the cell. I mean, it's a basic technical question that I know a lot of researchers who work in, gene therapy. It's just second nature. But could you explain some of the methods that you're able to deliver new genes or different genes into the cell?
Are you talking about for research purposes or for
Yeah,
therapies?
uh, for mainly for research, but for therapies as well.
Hmm. Well, it's a big difference. Like in terms of in research, when you're doing cell culture and animals, you're not too worried about like insertional problems about disrupting existing genes and such like that. You just want to get your protein expressed, and you're not trying to keep anything alive. You just want to show that it's functional. But when you go to actual therapies, there's a lot more to consider. I mean, obviously gene therapy has huge potential, but there's been some difficulties as well in terms of like having genes insert themselves into regulatory elements or other genes and cause cancer and that sort of thing. So those are issues that like every aspect of life extension depends on getting efficient, effective gene therapy. And for me, I'm more interested in focusing on figuring out what genes we'll want to use in the gene therapies once we have them, because there's many people working on gene therapy, and there's a lot of different methods that you can use to make it work.
I mean, I've heard.
Really
don't they use like viral vectors or something like that?
Yeah, there's adenoviral vectors. There's all sorts of lentiviral vectors. There's all sorts of them.
Okay.
And there's a like a new generation of more advanced ones that actually can do site-specific integration of genes. But it's yeah, it's a complicated area.
Sure, it's a complicated area, but
do a whole talk on that.
Sure, yeah, you could do a whole talk on it, but a lot of it has to do with putting the genes in like a virus, a dead virus, or a virus shell, or whatever you want to call it, and then sending that into the body, and then your cells take that virus in, and that's how the genes get into your cell, right? That's kind of a basic, one of the more basic methods, if I'm...
Yeah, that's it. And so you can add extra genes to your payload. Like, and sort of have extra proteins that will cause the DNA to be integrated into the genome in a specific place, and other things like that. So the basic strategy is to use sort of viruses that have had their the disease-causing elements removed.
Removed, right? Okay. And now I just wanted to say we covered protofection, and we also covered allotopic expression. Is there anything else on your radar screen that could help with MitoSENS that you've seen? I imagine that you keep up to date on a lot of different mitochondrial research and cell research, stuff like that. Is there anything else that you've seen out there that could possibly be of help?
Yeah, there's one other major technology that as an alternative to protofection and allotopic expression, and that is RNA import.
Oh.
So a lot of actually pretty much all organisms have import certain RNAs into their mitochondria, and it's recently been found that even in humans certain RNAs are actually imported into the mitochondria. And so instead of having proteins imported into the mitochondria, what you might be able to do is import RNAs. And because the mitochondrial genome is very small, it's actually smaller than most messenger RNAs,
Really?
you could import entire mitochondrial genomes using RNA import.
Okay. So that's
RNA versions of the mitochondrial genome, which would be continually expressed by the nucleus and sent to the mitochondria.
Okay. Well, that's good to know that there are a lot of different avenues out there to experiment.
Yeah, the interesting thing about cells is they're very complicated and they're often very redundant, so there's many different ways to achieve the same goal.
Yeah, I suppose that's kind of a good thing and a bad thing, that everything is redundant and complicated, right?
Yeah. The redundancy is good, but the complexity that comes with it can make it difficult to do your research.
I see a couple other people ask a question here. One about do you expect... I don't know if you're watching the chat at all.
Yeah, I'm just taking a look at it now.
Yeah.
There's a couple of questions there.
The one about yeast and worms and MitoSENS. I believe Age Vivo maybe is talking about doing allotopic expression, or maybe protofection.
Let me just take this. Do we have... do we have an idea of how much effect it would have?
Oh, I see what he's saying. Okay, yep, I understand now. Right. how much of an effect will MitoSENS